Good afternoon, everybody, and thank you to the organizer for inviting me and giving me the opportunity to tell you the story of the development of vaccines against Ebola and filoviruses in Janssen. I think you have all been fully aware of the large epidemic of Ebola in West Africa, which happened from 2014 to 2016. If you look at it by the numbers, clearly compared to many neglected tropical diseases we heard about this morning, it's really very few cases and very few deaths. But these emerging diseases are nevertheless important because of the public health threat they trigger, and also because some of them have a very high death rate, as you can see with Ebola. So they differ from neglected tropical disease because they are really making the headlines of the newspapers. They also mobilize stakeholders. In 2014, 2015, there was kind of a panic in the world, and every newspapers were talking about Ebola, about the fact Ebola was out of control, and it's a very legitimate fears when you have a disease that you don't know very well, which is susceptible to spread and which has a very high fatality rate. As you may have seen last week, we are now seeing Ebola back in the newspapers because there is a small outbreak in DRC. So as of today, it's a small outbreak with something like 20 cases. But as every time with this disease, it triggers mobilization and fears. Actually, these emerging diseases, even if they are very different from neglected diseases, they nevertheless share some features with the area you are working in. First of all, these diseases often affect populations which are really poor, having a very little access to any healthcare infrastructure, who are suffering for a very high morbidity and mortality level. And this disease can also trigger stigma and discrimination because of the fear of being infected by other people who are sick or who are survivors. Many of these diseases are happening in tropical, subtropical areas. I removed the point number 3, which was in the ISNTD definition by WHO, because as you will see with Ebola and Philo, we cannot say that they can be controlled, eliminated, or eradicated by any mean we perfectly know today. But I think it was finally the case also for many diseases we talked about. And then the last point is relatively neglected by research. And actually, I think it was the case for Ebola before 2014, even if some research was ongoing, probably not enough. And this can happen again because there is a competition among emerging diseases, and it's relatively difficult to know which threat will be the next threat. So the focus can move from one disease to another disease. And this does not go well with research. In research, you need a lot of perseverance. So the research for Ebola and Philo dramatically accelerated in 2014-2015, and this was really thanks to the mobilization of all stakeholders. So when I say all stakeholders, it was the public health institutions, it was the industry, and it was also the funders and the governments. So now what is important is not being distracted by other emergencies and finishing the Ebola job. I will be telling you about our Janssen Filovirus vaccine program. Since the epidemic in West Africa, there are quite many companies working in this field, I think a dozen. What happened in Janssen and why we have been able to embark in this adventure, it is that we were having early research on Ebola. Actually, we were already having a program which was funded by NIH to develop a multivalent filovirus vaccine. And it's thanks to this program that we have been able to accelerate our research. And because a multivalent vaccine is always more complex to develop, we decided first to focus on a monovalent vaccine. It's how the story started. So here what I would like to tell you is that you cannot start from scratch when there is an emerging disease. And as a company, you can rely on some internal research, and here, fortunately, it was the case. You can also rely on academic research and partner with academics. But you cannot, decide that you need the fireman when the fire is already here. So these early discovery programs, even for threats which are very uncertain, are extremely important. From 2014, we decided to accelerate, and we decided to develop a heterologous prime boost vaccine. It means it's a vaccine with 2 doses, and it's 2 different vaccines. One is from Janssen Research, it's an Ebola Zaire adeno-based vaccine, and the other one is from our partner Bavarian Nordics, and it's a MVA Bavarian Nordic filovirus vaccine. And from 2014, we have started to study this prime boost vaccine with different intervals and different sequences. So as you can imagine, it's relatively complex to move forward in clinical development. And relatively quickly, we also realized late 2015 and 2016 that we had also to continue with our multivalent program. And now we have a phase 1, which is ongoing with the multivalent program. So we had to focus on 2 different things, finally, a multivalent vaccine and a monovalent vaccine. As I said, part of our vaccine, the Bavarian Nordic MVA comes from Bavarian Nordic, and the other part, the adeno 26-based vector vaccine, is coming from Janssen Research. So partners are extremely important. Some of them are industrial partners like Bavarian Nordic, but we also had a lot of support from the European Commission, and this came in the format of an IMI project. And we have we are running actually 4 IMI-supported projects. And here we have some funding from the European Commission and plenty of partners working with us who are either academia or NGOs or other companies. In our research, we also have support from US funders like the NIH and also GVAP or BARDA. And we also partner with some people who are providing us more technical support, especially in some countries like the Walter Reed Army Institute of Research, which is acting in some countries in Africa, or the HIV Trial Network. So there is an incredible value in partnering if you want to move forward this type of vaccines for technical aspects and for funding aspects. So what did we do since 2014? We started in a very short timelines 10 clinical trials. The phase 1 are completed, but the phase 2 and phase 3 are still running. Some of the phase 2 are almost completed, but because of the regulatory process, which is not fully cleared yet, they are not even unblinded, so we don't have the data yet. This represents more or less 2,000 subjects vaccinated by one of the 2 components of the vaccine or both. And recently we had a new trial which started, which name is PREVAC, which is sponsored by 3 partners. So it's a massive clinical development program. Usually it takes many more years, but there the 10 trials were really started in very short time frame. We have already knowledge about our safety. So today our vaccine regimens have acceptable safety profile. And I say regimens because the safety is the same whatever the interval between adeno 26 and MVA is. And also you can have different sequences, adeno 26 followed by MVA or the opposite. And in any case, the safety is the same. We fully know the safety from the 4 phase 1, so this vaccine triggers local and systemic reactogenicity. It's mainly pain at injection site and also some fatigue and headache. And in phase 1, we did not have any serious adverse events related to study vaccines. Phase 3 and phase 2 and phase 3, as I told you, they are still ongoing. There has been more or less 2,500 persons exposed either to vaccine or to placebo. It's still blinded, so I cannot share with you a full picture of the safety data from phase 2 and phase 3. So from blinded database, vaccines are generally well tolerated today. We had a temporary clinical hold during the development. It was last year. We had to hold for some months our development plan to further investigate 2 serious adverse events. One was post-MVA. It was finally considered as not related to the vaccine dose, and another one was post-adeno 26, and it was considered as doubtfully related to the vaccine after investigation. So this hold was subsequently lifted, but we lost several months, and it's very legitimate to go through a very thorough investigation. Now the clinical studies have restarted in all countries except one. We already have some documentation of the clinical immunogenicity of this vaccine. So we have shown that with the vaccine we trigger strong binding antibody responses. And we have shown this both in the US, Europe, but also in Africa in phase 1. We trigger binding antibody response after the prime, and then when you give the boost, it's substantially boosted with all regimens. What we saw is that with the regimen with 2-month interval, the boosting effect is stronger. And we also had the possibility to follow up subjects until 1 year post-prime. So we see a good persisting immune responses up to 1 year. There is a steeper decline in Africa, but nevertheless the immune response is persisting. We have also shown that on the top of binding antibodies, we can show we have neutralizing antibodies, and we have also T-cell responses. This was published so far. In terms of manufacturing, because of the public health threat context, the company decided to manufacture large amounts of these vaccines at risk. In general, when you are in preclinical or in early phases of clinical development, you manufacture moderate quantities of vaccines, the quantities you need for your clinical development, basically. And then when you manufacture further batch, it's to go through your process validation. But there, the company really decided to take a big risk and to manufacture large quantities to help addressing this outbreak in West Africa. So we have 2 million dose regimens of drug substance, and we have 1.6 dose regimen, which are already in vials. They are released and ready to be used. We also worked on the stability profile, because it's important to be able to use the vaccine with the cold chain and what is existing in terms of equipment in Africa, and also to use it in real field conditions. So we studied the stability at +2 to +8 degrees, which is normal fridge. So far we have 9 months of stability for Adeno 26 and 6 months for NVIBN Philo at these temperatures. And at -20 degrees, we have longer stability profile. So these data are preliminary because month after month, we know more about stability, but it's not something you can document in very short timelines. So we'll probably have even longer stability periods moving forward. We also made some experiments to see how the vaccine behaves in real field conditions. So we did experiments with agitation, freeze-thawing, and also exposure to 40 degrees for a very short period because you have some time between you take the vaccine in the fridge and you put it in the syringe and you inject it. So you have a short period where the vaccine can be exposed at high temperature. So we also studied that. Another part of our program was building community trust. Among the clinical trials, one is in Sierra Leone. It was initially conceived as a very large trial. Finally, it's a large trial. In Sierra Leone, the context was extremely difficult with a population which was suffering a lot, which was having absolutely no trust in the healthcare system, no trust in the government. And we were thinking that proposing a clinical trial on an Ebola vaccine in this country could really be very poorly perceived and really rejected by the population. So we were already facing this issue of trust, and we were facing another issue, which was an important issue for a clinical trial. It was the fact that people were not having any personal ID in Sierra Leone. And when you have people in clinical trials, you need to know who is coming and who is coming back for the following visits. And also because the vaccine is with 2 injections, the prime and the boost, you want absolutely to be sure that the person you're giving the boost is the real person you gave the prime few months or few weeks ago. So we also developed a biometric identification tool to be able to carry out this trial in Sierra Leone. And we also used messaging to the mobile phone of the people to encourage them to come back to the visits. And actually it has worked quite well. It was quite a lot of investment because we really engaged with every level of the community, the governments, but also the regions, the district, the traditional leaders, the religious leaders, etc. We organized a lot of household visits and etc. To be sure we were spending enough time with people who were absolutely under stress because of this Ebola epidemic, who were absolutely not used about clinical research and who were, asking questions, and it was very, very legitimate questions. We hope that all this effort will have some legacy for the country, and that there will be some more capacity in the future in the country to conduct further research or to address other health issues or health crises. So it looks like we have done a lot in 2 or 3 years. Nevertheless, we are far from having a vaccine with licensure, with an approval by any regulatory agency. And we don't even fully know today if and how this will be feasible. First of all, I have shown you safety data and immunogenicity data. But when an epidemic is over, you have no more cases, so you have no possibility to demonstrate clinical efficacy. So obviously, it's very good for the populations, but basically for the development of a vaccine, you cannot bring the evidence of the clinical efficacy. So this is a question mark, and you have to go through different regulatory pathways, which are not the classical regulatory pathway, and especially you have to use a lot of non-human primate data. But this is not very easy because it's really new for us, but also for the regulators. Then the mobilization of stakeholders towards Ebola has decreased, and there has been other health priorities in the world. We also now face a funding issue because we had initial funding to fund our initial program, but today we don't know what will be the funding in the future. We think also it's not sure that anybody in the world, any country, any region will ever need big quantities of these vaccines. The last outbreak was very large in West Africa, but usually the Ebola outbreaks in the past, they were very small, so we don't know about the volumes that will be required. And also, as I told you, there are several companies developing vaccines, and one company is more advanced than we are, and it's the RVSV vaccine from Merck, and they have already demonstrated efficacy before the end of the epidemic, and they are also now recommended by the WHO SAGE. So this is just to explain you that we are at a turning point as a company, and we continue and we want to fulfill all our commitments, but the path forward is not yet fully clear for us. To make the things more complex, you also have other filoviruses in Africa, and as I told you, we have now resuscitated also our multivalent filovirus program. So despite significant achievements, the future is not fully clear, and we hope we will be a potential contributor to help preventing or mitigating future Ebola filovirus outbreaks. At least it's our ambitions, and we have already done something in this way, but it's not the end of the story. Actually, we think that to continue working in this field of emerging diseases, we need new ways of working probably, and even if we have worked with many partners for Ebola, we probably need new type of collaborations or even more public-private partnerships, because there are technical issues to develop these vaccines, like the unknown regulatory pathway, the absence of demonstration of efficacy, but there are also business challenges. We have a big inventory. We don't know what will be the medical need for this vaccine. We don't know what will be the business model. So it's not something that companies can move forward alone and in isolation. I'll give you an example of a new coalition where we place a great hope, and we are very happy to be strongly involved and to be part of this coalition. It's the CEPI, Coalition for Epidemic Preparedness and Innovation. And the ambition of these stakeholders is first to finish the Ebola job, but also to develop new vaccines, at least until phase 1 or phase 2, to react faster if we have new emerging threats, and also to develop some vaccine technologies or vaccine platforms that could then be accelerated in case there is an emerging threat happening again. So it's the type of thing where really place some hope. And yes, as a conclusion, I just want to acknowledge and to thank all the people who are supporting us in this adventure, including the volunteers in the clinical trials and the investigators. Thank you.